So what does happen when a base has been changed by deamination? Due to the chemical alteration and the fact that amino groups are actually really important in forming hydrogen bonds with the other bases and participate in base pairing, the chemical change due to deamination now will change the base pairing between the altered nucleotide and the sister DNA strand. And as a result, if the error is not caught before DNA replication occurs, in the next DNA, after the next DNA replication, a mutation will have been incorporated into the DNA. So let's look at what actually happens. So cytosine was reacted with water that's all around the DNA, and a deamination has occurred, changing cytosine to uracil. Now, normally cytosine base pairs with guanine, forming 3 hydrogen bonds between the bases. But since the deamination has changed cytosine to uracil, next time the DNA is replicated, the DNA polymerase, which will select the next base to incorporate based on the base pairing, will not incorporate a guanine in that location, but it will incorporate an adenine, which efficiently base pairs with uracil, unlike guanine. As a result, after the next round of DNA replication, there will be a G to A point mutation in this location. So cytosine is not the only nucleotide that can be changed by deamination with water. Adenine can be changed to hypoxanthine, and guanine can be changed to xanthine. Now, guanine to xanthine change is the least troublesome because both guanine and xanthine will base pair with cytosine. So even if this mistake is not caught by the DNA repair machinery, there will be no mutation in the next DNA replication cycle. However, when adenine is deaminated to hypoxanthine, it's the hypoxanthine molecule will no longer base pair with thymine, but will form 3 base pairs with cytosine, resulting in a T to C point mutation in the sister strand. So deamination is the rates of deamination are reasonably low inside cells at physiological pH. However, the cytosine to uracil deaminations is much more frequent than the other ones. It's 100-fold more frequent at the physiological pH. So the cell has machinery to fix these errors. And here, let's pause for a second and ask ourselves a question. So pause this lecture, read the question, think of what you would select as an answer, and then I'm going to give you the answer. And you can see if you were right. So the answer to the question is, of course, C. When a deaminated cytosine is changed to uracil, one DNA copy will be normal. That's the bottom copy here that still has a guanine there. And the other will be mutated. This is the copy that has a uracil. And if not fixed, and if this DNA strand is allowed to serve as a template during next DNA replication, it will result in a permanent mutation incorporated into the DNA. So the machinery, DNA repair machinery responsible for fixing these deamination errors, as well as a lot of other errors inside that alter the bases, is the base excision repair proteins. So base excision repair, the actual word implies that first the base of the nucleotide is removed, and then the rest of the nucleotide is removed and replaced. Both in prokaryotes and eukaryotes, the enzymes responsible for that are glycosylases. It's a whole family of enzymes. In this case, because we're looking at the cytosine change to uracil, it's going to be the uracil DNA glycosylase. So this enzyme has the capability of both binding DNA and sliding along of the DNA, recognizing a deoxyuracil inside the DNA molecule, and clipping out, glycosylating, breaking the glycosyl bond that attaches the uracil to the ribose, the sugar. So the name of the enzyme actually describes really well what it does. It cleaves the glycosyl bond, creating a basic area. So you guys can see that the base pair has been removed, but the backbone, aka the sugar and the phosphate, have not been altered. These particular abasic sites are then recognized by a different enzyme, which is the AP endonuclease. AP stands for apurinic or apurimidinic endonuclease. So either the purine or the pyrimidine in this particular location is missing. So what it does is then cut out the sugar and the phosphate from their basic site, leaving a nick in the DNA, which has a 3' OH and a 5' phosphate. So as a result, now either in both cases, a polymerase can come in and then either fix this nick right away by inserting only one nucleotide in this location and then ligating it in, or by removing a short chunk of DNA and then repairing it by a ligase or an endonuclease. It will remove this extra flap of DNA. So there's another question I want to give you. You guys should read the question, and the question is, which is the correct product after AP endonuclease activity? And pause this video, think for yourself what it would be, and then I'll give you away the answer. So the answer is B. This is the only version of these nucleotides that has a 3' OH, and that's what the DNA polymerase needs in order to extend a strand. So B is the correct answer. The 3' OH of the ribose is all the DNA polymerase needs. So that's what AP endonuclease leaves it. Now, we just talked about how water damage is repaired. Deamination by water is repaired by base excision machinery. Now let's talk about how replication errors are corrected that happen when DNA polymerase makes a mistake. Now we know that DNA polymerase already has its own proofreading activity. Most DNA polymerases have a 3' to 5' endonuclease site, which will remove the offending base pair. And it can remove it because most incorrect base pairing will create a bump in the phosphor-sugar backbone of the DNA, putting this bump a bit closer to these two magnesium ions, which will then catalyze the removal of that nucleotide from the location, simply because the base pairing is incorrect. So you guys can see in this slide that you have seen before is that it's the barren shape of the incorrect base pairs that doesn't quite fit into this exonuclease site, placing the base pairs close enough to the catalytic magnesium to break the phosphodiester bond. However, this exonuclease activity has to be extremely fast because our DNA polymerases are so fast. They can catalyze hundreds of bases per second when they replicate DNA. In fact, they have to because there's so much DNA to replicate during DNA replication of a eukaryotic genome. So mistakes are made. And when mistakes are made and an incorrect base pair is incorporated, despite the fact that it leaves a little bulge on the DNA backbone, the mismatch repair machinery will fix those mistakes. So you guys can see here that this is the correct base G, and here the DNA polymerase, while replicating the DNA, has incorporated by mistake an incorrect base and then did not proofread it, but moved on, leaving an A here instead of the proper C. If left unrepaired and the next cell division is allowed to occur, so the DNA gets to be replicated again, this lighter blue strand will serve as a template for DNA synthesis and will result in an incorrect base pairing on the new strand, because that's all DNA polymerase can do. It sees an A here, well, it will incorporate a T. And as a result, this half of the cells, one of the daughter cells of this mutated cell will have an incorrect strand, and the other one will be correct because it used the correct DNA template as correct DNA strand as a template. Well, the cells can't have that. So they have these little super enzymes called DNA mismatch repair. And they do their hard work to try to repair these little bulges after the DNA replication machinery made a mistake. In fact, they follow really closely with the DNA replication machinery. So as the DNA is being replicated, it also is being repaired. And we're going to see why in a second. We're going to see why now. So if the DNA mismatch repair has a conundrum. It knows there's a mistake here because there's a bulge. The DNA mismatch repair sees that there's incorrect base pairing going on, but how does it needs to make a decision? Which of the two strands it will now use as a template, and which of the two strands is incorrect? How does the DNA machinery? It has to be able to recognize the old strand, which is most likely correct, and then the new strand, which is most likely was polymerized incorrectly and incorporated this mismatch error. Because if it happens to choose the new DNA strand, it will then propagate the error to the next generation, because it will have corrected the bulge, and instead of fixing the A, it would have fixed and clipped out the G and put a T in here to make sure there is no bulge, and now we have a mutation, a point mutation in this location. So that makes DNA repair machinery sad. However, if it chooses the correct template strand, so if it realizes that G is what should be there and A is what's wrong, it will replace the A with the C, and then there's no mutation in either of the DNA strands upon next cell division. So how does DNA machinery, mismatch repair machinery do that? What is it about? How can it recognize new DNA? Well, turns out that for a little bit after the DNA has been replicated, the new DNA is still full of nicks. So the DNA ligase is not as fast as we would like, and so for quite a bit of time after the green DNA has been made, it still has all these 3' to 5' nicks that need to be repaired by DNA ligase. Because there are nicks in the new DNA but not in the old DNA, the mismatch repair specifically recognizes the nicks first. And only when it has found a nick, it orients itself onto that strand and excises that part of the strand as the new strand and the strand likely to have the incorrect base. So here you guys see it. So if you zoom in, for example, this location, there's a nick in the DNA, and that's what mismatch repair machinery needs to realize that this is the new and this is the old. So as a result, mismatch repair has to occur shortly after DNA replication has started, so that the DNA ligase does not have a chance to repair all of these nicks and to make it impossible to recognize which DNA is old and which DNA is new. So here's the repair process. There's a DNA mismatch, and only the red newly synthesized strand will have a nick in there. The mismatch repair machinery involves two enzymes that can bind to each other, and one of them recognizes the mismatch bulge, and the other one recognizes the nick in the DNA strand. And because one of the enzymes binds the nick and the other binds the bulge, the way they come together will result in in the excision of only the newly synthesized strand, but not the old correct strand. This empty chunk of a single strand of DNA is then repaired by DNA polymerase, and breaking sites, the nicks of the repair right here, will be repaired by DNA ligase. So the eukaryotic and prokaryotic DNA mismatch repair machinery is quite similar, at least between yeast and E. coli. However, much less is known about how mismatched DNA repair occurs in humans. So here's the laundry list of the enzymes that participate in mismatch repair in a simple eukaryote, so yeast, and then a complex one, a human. And if you compare it to the DNA mismatch repair of a prokaryotic cell, they are very, very similar. They're similar proteins that fulfill very similar functions. So MSH2 and 6 are the mismatch repair proteins that recognize recognize the bulge and the new DNA nick, and then exonuclease 1 and RPA fix. Exonuclease 1 removes the newly synthesized strand, and RPA coats the old strand, preventing secondary structure from arising inside the template, and then DNA polymerase will come in and synthesize new strand of DNA, hopefully correctly, based on the orange template. So, because there are several levels that are built into DNA repair to assure that DNA repair occurs, DNA replication occurs as precisely as possible, the final error rate of the DNA replication is only 1 mistake to 10 to the 9th nucleotides, which is very, very low. DNA replication without proofreading, just how the DNA polymerase incorporates nucleotides as it reads the template strand, is far lower. So there's orders of magnitude increase, 100-fold here and then 100-fold there, to achieve such a fantastic, such a precise DNA replication that DNA polymerases are able to provide us, which is very good, because even though not all mutations as they arise are deleterious, it's still very, very important to make them as few as possible. However, a certain DNA mutation rate due to DNA replication is also important. And that's rarely appreciated when we talk about how, the bad mutations occur and how they're not good and how we must fix them. Because it is the low DNA mutation rate of DNA polymerases is also still a driver for evolution, allowing for the possibility of change. Even though some of the change will be deleterious, there's still a potential that some of the change will be an improvement that will allow the organism to adapt better to its changing environment, because nature is never static and things change. So it's important actually for ourselves to have a certain error rate, and it's actually not a bad thing that the DNA replication error rate is kept very low, but it has not been completely eradicated from ourselves. Now let's talk about another aspect of DNA damage and how it can be repaired by our cells. And in this case, it's the ionizing radiation, so X-rays and radiation. Ionizing radiation, because it's so energy rich, destabilizes the backbone, breaks the phosphodiester bonds of the backbone of DNA, and can result in double-stranded breaks. This DNA type of DNA damage can be repaired by 2 different mechanisms. One of them is the non-homologous end joining, which is the, oh my god, my DNA is broken, I must do something or I will die. My chromosomes have just been completely fractured. Let me tape them together as best as I can. So that's basically the gist of non-homologous end joining. The cell has realized that it has broken chromosomes that are not attached to anything, And it does its best. It trims off the break by nuclease and then it glues them together somehow. That is not the preferred way to repair such breaks, but it is quick and dirty and it gets the job done. It also is one of the ways to cause indel mutations and chromosomal abnormalities inside your chromosomes. It is activated in single-cell organisms such as bacteria or yeast after a massive exposure to X-rays. But we also have non-homologous end joining in our cells, which is not. We prefer a different type of DNA repair. But it's important to know that because non-homologous end joining removes some of the DNA molecules in order to create this smooth cut that it can then rejoin using DNA ligation, there will be lost nucleotides in this mechanism. The other way that DNA can be repaired from double-stranded chromosomal breaks is homologous recombination. It's also a very important mechanism that we use to create transgenic and knockout animals for research, and hopefully I get the chance to talk about that when we talk about recombination later. So this is sort of a preview. So when there's a single-stranded nick inside a chromosome and replication is occurring, it can result in replication fork collapsing and one daughter chromosome being shorter than the other. So damage like that has to be repaired. So if you now look at this two uneven daughter chromosomes and turn them on the side, you'll get something like that. So there's a shorter chromosome that had a nick here and was broken off, and here's the longer one. So what happens first, an exonuclease will come and chew off 5' to 3' some of the parent template strand, leaving a 3' OH here on the synthesized daughter strand. Then the recombination machinery comes in and it flips the two synthesized daughter strands, allowing for the second daughter strand to use the blue template to be fully extended. While this long daughter strand is used to re-extend the parent strand of the blue molecule of the upper DNA of the upper chromosome, and this allows the new replication fork to be formed in these locations and for DNA to be repaired using the correct template of one of the chromosomes. The complete template of the old chromosome can be recovered. The orange things here are the Okazaki fragments forming. There's still a 3' OH that of the old strand, and this guy is going to be extended. And this X structure, as we're going to learn when we talk more about recombination, is then reformed again correctly using an enzyme called resolvase. And the big benefit of the homologous recombination mechanism that is used to repair double-strand DNA breaks is that there's no lost nucleotides for this mechanism, but one of the sister chromatids serves as a template to repair the other sister chromatid. Now, the different DNA repair mechanisms that we have discussed today are only a few of the many ways that our cells have evolved, of the many mechanisms our cells have evolved to repair their DNA, because it's so important for our cells to make sure that DNA is replicated very precisely. Yes, a certain mutation error rate is great for evolution, but for the most part, it's better when the DNA is repaired. And only viruses like to have a very, very high error rate because they're so simple. So this is just a summary of different DNA repair mechanisms that exist, so you guys can appreciate the complexity. Not that you need to memorize every single one, and by God, don't even think about memorizing the names. And you guys, if I'm not sure if it comes out here, I mean, it doesn't, but a lot of the DNA repair mechanisms are really when those proteins are mutated themselves, is one of the reasons why we get cancer. For example, BRCA1 and 2. So two proteins play a role in recombinational repair in eukaryotic cells are the key indicative genes for breast cancer. So BRCA1 and BRCA2. If you have a mutation in this gene, you have your homologous recombinational repair is not working correctly, and you have a much higher chance of developing breast cancer. So here's a summary of things we talked about today. Have fun reading it and using it for learning. And then also, there's another TED video you guys can choose to watch, which is called "What Happens When Your DNA is Damaged." It talks about DNA damage and repair on a much more basic level that I covered today in class, but I think it makes for a very nice and stylish summary of this lecture.